I'd like to thank the organizers, Andrika and Grace, for this opportunity to talk at the conference. Unfortunately, I'm right in the middle of moving my laboratory between two sites in New Jersey, so it was difficult for me to be there. I was very excited to see that astronaut Joe Abaca was at this meeting today. Joe was here at Merck in New Jersey for a NASA day in 2016. And everybody was so impressed with his presentation and the time that he spent to talk to multiple researchers here at Merck. So let me just start off with, go to the next slide. Okay, so my day job is I'm an associate principal scientist at Merck Research Labs. My main function is looking at opportunities for transitioning biologics, in particular monoclonal antibody drugs, from being IV to sub-Q formulations. And one of the things that we're working on is the idea of using crystalline suspensions. We're also interested in seeing if crystallization can be used for manufacturing applications. I started my career, believe it or not, almost 50 years ago. I was drafted in the Army. So I spent 2 years working for the U.S. Army Chemical Warfare Center. After I graduated college, since then I've spent 45 years in the pharmaceutical industry. I've had the pleasure of doing 16 microgravity experiments, 12 on the space shuttle missions, and now 4 going to the ISS National Lab. I have 5 kids and 2 grandkids, 4 cats and 3 dogs. But only 1 cat and dog is actually mine. My kids tend to come and go, but the dogs and cats stay. Why is everybody interested in crystallization? So for small molecule drugs, like Januvia, insulin, ibuprofen, aspirin, they're all crystalline materials or use crystallization in their final process for manufacture. However, compare the size of Januvia and insulin to a monoclonal antibody, you can see the size, not only the size, but the complexity of the molecule is tremendous. The other difference with monoclonal antibodies is they're not rigid, they're very flexible molecules, so this remains a challenge to crystallize those. So what we're trying to do is to extend the knowledge that we've gained in the pharmaceutical industry with small molecules to biologics. And one way we're hoping to help us come up with crystalline suspensions is to use microgravity as a research tool. So let me just start with what's usually done in the manufacture of monoclonal antibodies. Usually it's chromatography, chromatography, chromatography. These are high-cost steps. The one step in particular, the protein A chromatography step, is 30% of the manufacturing. So we're looking for what's used in small molecule processes, green and sustainable processes, where you simply crystallize your substance, your drug substance, you filter it away from impurities. This is a simple step. It would lower cost and reduce waste. There's a need for high concentrated monoclonal antibody formulations. Currently, right now, most monoclonal antibody drugs are given as IV infusions. So this generally involves taking the patient going to the hospital. There's multiple steps in preparing the person for the infusion. So there's a 1-hour infusion, saline infusion. There's the actual monoclonal antibody infusion with a follow-up to wash out any remaining drug that's in the lines. So that's a total of 3 to 5 hours of time. A large percentage of the patients that are on monoclonal antibody patients have implants, and the reason for that, they have these ports put in because they don't have the vascular system to support a IV infusion. And generally, this delays the administration of the therapy, and there's always a concern about infection using these ports. So what we're looking at is seeing if we can transition from these IV infusions to subcutaneous injections, where you can take 200 milligrams per ml of a crystalline suspension. That could be done in a doctor's office in 5 minutes. It would eliminate the cost, actually 50% of the cost of administering the drug, and it would improve the adherence and compliance and really the quality of life, not only for the patient, but normally, the cancer patients that are taking these therapeutics, caregivers are taking time off from their work and their vacations in order to accompany their patients to these visits. So why don't we just move over from IV to sub-Q? The issue is viscosity. So as you increase the concentration of the monoclonal antibody solution, we find that the viscosity goes up dramatically outside the range of what would be suitable for an injectable product. However, it's been found that in the case of infliximab, which is the active ingredient in Remicade, that the crystalline suspension remain even at 200 milligrams into an acceptable range of viscosity for crystalline formulations. So what does this have to do with microgravity? So what we're looking at is taking advantage of 3 microgravity enhanced crystallization effects. If you look at the image on the right, that's a picture of a crystal that as it's growing, because of its mass, it's sedimenting in the solution that it's growing in. And you can see the trail of particles that are following it. So what this results in is particles that have less high quality than they would if they could remain stable in a microgravity environment. In the panel below, you see the same crystal growing in microgravity, and you can see how the molecules can just march into that crystal and improve the overall quality of the crystal. The second effect that we'd like to take advantage of is that molecules just move more slowly in space. So we're hoping is that this slower diffusion may enhance the opportunity to grow more perfect crystals. The third effect, which is actually my favorite, is using temperature effects. It just so happens that in microgravity, that convection is a gravity-driven process. So the best way to demonstrate that, if you have a pot of water or a clear vessel and you're heating it from the bottom, what you can see is that swirling that's taking place in that vessel as you're heating it, those are convection currents. That's an indication that there's very different temperature gradients going on within that vessel. However, if you do that same experiment, microgravity, you get uniform temperature throughout layers of that vessel. And this gives you an opportunity, if you have a process where you say the crystals only grow at 11 degrees, you can get thin layers of crystals growing very evenly throughout that vessel that you have. So overall, we're looking at whether microgravity can give us higher order crystals, which are more pure, and in the case of my application, more uniform suspensions. So how does a researcher go about doing microgravity experiments on the ISS? So I work with the ISS National Lab organization, and the first step in that process is to make a proposal. And one of the key things that you have to show in that proposal is benefit of life on Earth. And usually what it involves is using flight certified hardware That means basically hardware that was developed in the space shuttle era that you have to use for these experiments. The next step, if you get a successful proposal, you have to do a pre-verification step, and you have to demonstrate your experiment feasibility. You lock down your experiment protocol, and then actually comes the time, which we're very excited about, we actually load our experiments into the hardware, and a very happy event takes place called turnover. That's where you turn over your experiment to NASA, ultimately turns over the experiment to SpaceX to load onto the Dragon module. After launch, the Dragon separation, the Dragon rendezvous with the ISS, and then after that, your experiment is usually stowed until it's activated. During this, usually we do the experiment in what's called cargo resupply missions. So these usually are about a month in time. So that's the time that we have to get our experiment to happen. And then upon recovery, once we get our sample back from the Dragon module, that right now is landing back off the Gulf of Mexico. And believe it or not, my last experiment from the time that the Dragon parachuted down in the Gulf of Mexico, I had my experiment back in the laboratory in Merritt Island within 2 hours. And then once we get our samples back, we perform a battery of microscopic and biochemical analyses. So most of my experiments aboard in collaboration with the ISS National Lab have been done with pembrolizumab. Pembrolizumab is the active ingredient in Keytruda. Just to give you an idea of the cycle that takes place for an experiment, we start off where we load our experiments into two types of hardware. One is called the protein crystallization device, and then there's the protein crystallization facility. This allows you to get crystals that are suitable for structural work. These towers are individual 1 ml bottles, so there's 35 bottles that are in this tube. So you have an opportunity to have 35 experiments in one of these tube experiments. Our experiments go up into what we call Merlin incubators. In our case of our experiment, that's kept at 4 degrees. Generally, it can take up to 4 days to actually get the Dragon module to the ISS. And generally, we have about 18 days of actual time for doing our crystallization experiments. Then the experiment goes back into a storage bag, and then within 1 to 2 days, we have our experiment back for analysis. So how'd we do? All right, so for SpaceX 3, which was in 2014, one of our monoclonal antibodies, we were interested in doing structural studies. This is the hardware that we used. This is a photomicrograph of the ground experiment and the comparable flight experiment. And right away, you can see the difference that in the ground experiment, we have nice crystals. However, the flight crystals are not only larger, but more of the monoclonal antibody has crystallized. These crystals diffracted better in X-ray diffraction studies than the comparable ground experiment. So we were very happy with that result. On SpaceX CRS-10, which was in 2018, we were doing experiments with pembrolizumab, and this is primarily for drug delivery. So this is the hardware that we used, and we use a technique called sonic. It's a very fancy name, second order imaging of chiral crystals. So this basically allows you to show if you have crystals within the particles that you've generated, whether they are crystalline. We confirm that the particles are protein by looking at the particles under UV. And then we use a technique called second harmonic generated imaging, where we can tell whether those same particles are crystalline or not. So one of the things that we found, which was a total surprise, is that instead of getting large crystals like we thought we would get in this experiment, we got a suspension of really small crystals of uniform quality and size. So this gave us an excellent opportunity to look at, you have one preparation that has this heterogeneous population of crystals, the other one has this tight population of small crystals. Does it make a difference in an application like drug delivery? And what we found is that the flight experiments, the small uniform suspensions, had better, lower viscosity and better injectability properties than the comparable ground experiment. And we used this finding to improve our processes on Earth. Currently, from this experiment, we came up with a process to mimic these results, and this is what we've used to prepare a preparation that's ongoing development as a sub-Q formulation. Let me just talk over what this slide was supposed to show. The slide was supposed to show that astronaut Kate Rubins, who in 2016, who was a molecular biologist, all astronauts are allowed to bring up one personal item. So what Kate did is she brought up a bunch of pipetters, and she just started pipetting in space. And when I saw this, it just blew me away, because for the first time I realized you don't need all this very hardened hardware that we were using for the experiments, that basically you can do pretty much the same experiment in microgravity that you can do on Earth, as long as you touch the surface of the vessel that you're transferring your material from. So this led us to our next experiment that we did in 2021. And what's unique about this experiment is that we made a 3D printed platform, which we could put on one side our monoclonal antibody solution, and the other side a precipitant. We did this in triplicate for a number of different platforms. We call this a platform. And this platform, we looked at purification, diffusion, and temperature effects. This is astronaut Mark Van de Heij activating our experiment. So basically, very simple experiment. There's a valve in between the two solutions. All Mark had to do was turn the valve to the up position and allow mixing between the two syringes. And then once done mixing, put all the solution on one side for the crystallization experiment to start. And then generally we do a battery of different tests to test the quality of the material that we get back. We do biochemical analysis. We looked at crystallinity. We looked at the particle size distribution, and then the bioactivity. And basically we found that the 3D printed hardware and overall experiment processing performed nominally, meaning that there were no issues. And we see this as an opportunity for first-time users to think about doing relatively simple experiments on Earth and easily be able to translate them into something that could be done in the microgravity environment. So in the case of this experiment, crystalline suspensions were derived in all the flight and ground experiments. We were able to confirm that. One of the interesting things we found that the less pure samples that we had in there, the purification samples, seem to benefit more from the being in microgravity than the ground experiment, and suggesting that diffusion effects would be important for improving the purification process. And finally, the other thing that we found was, one of the things that puzzled us is if you have a crystalline suspension of pembrolizumab, and you heat it up to 40 degrees C, it turns clear, and this puzzled us what was going on. So we performed the same experiment in microgravity, and what we identified this phenomena as what's called Oswald ripening, where small molecules melt and they form bigger crystals upon temperature shifting. So this was a great finding for us. So what do I see as the best use of the ISS for the remaining time for the end of life for the ISS? Is to see whether we can discover and develop processes which can benefit from microgravity effects. Especially, I think there is opportunity for doing small molecule crystallization and using seeds from those small molecule experiments for ground experiments. The other area that seems to be tremendous interest is in the area of nanoparticles, and for vaccines. So what we're hoping is that we can encourage other people to consider, researchers, to think about simple 3D printed hardware for proof of concept experiments, and the use and allowing for more flexible implementation from partners doing hardware design. So what are options post-ISS? I'm sure you're hearing about plans for future floating commercial space stations, Axiom Space and Sierra Space. However, one of the questions that always comes up is who's going to pay for the cost of doing experiments in microgravity? I think that there's an existing model that we used in our structural studies. We formed a consortium with other pharmaceutical companies to share the cost and resources for synchrotron facilities. I see this as a model for opportunities to share laboratories post-ISS. And finally, I feel that we're only in the infancy of doing microgravity research. I see that there's a need for doing considerably amount of research and development, and for doing an opportunity to do iterative experiments as a way to pave opportunities to have manufacturing applications in microgravity. Thank you.